Hey guys, I'm going to be running you through a presentation over the genome assembly and annotation of Idiomarina loihiensis by me, Casey Norman. And over here to the right, we have an electron microscope image of our organism itself, just to give you an idea of what we're working with. So the first thing we really need to ask ourselves here is, why are we interested in this? Why do we care? Well, Idiomarina is a very common organism found in marine sediments, and the extracellular electron transport capabilities of it are not entirely known. And just to mention what extracellular electron transfer is, it's the process by which some microorganisms exchange intracellular electrons with an extracellular electron donor or acceptor, including naturally occurring metal compounds and artificial electrodes across the cell's plasma membrane. And also, the potential biotechnological uses of it. These organisms have the potential of energy generation, wastewater cleanup, and more believed to be possible. And taking this through a genomic approach, we hope to better understand the processes that it can undergo and learn further capabilities of it. I just wanted to include this image here just to visualize what this extracellular electron transfer is. So starting where metabolism is in the center, that's going to be where we are intracellular, and then following the red arrows out towards the electrode, it's going to be moving the electrons extracellularly, just to put it more simple. And coming into our research questions, we just want to know, what are the amino acid metabolic potentials of our organism? What genes does it have, suggesting it can live and grow by utilizing certain biochemical pathways? And given the organism can successfully grow on a cathode in our lab setting, how will this extracellular electron transfer help us understand the electron uptake capabilities of this organism? Coming into our quality assessment of this, we had a 2.8 megabase genome, which is relatively small compared to really any other genome. We were also able to identify we had 2,644 protein coding genes, which gives us a lot of room here to work with in research. We found an average nucleotide identity at 95%, and using the Microbial Gene Atlas, we found our nearest relative to it being this strain of Idiomarina, coming in at 97.3% ANI. Our guanine cytosine, or GC content, we found at 47.02%, and greater than 40% really suggests that's a reassuring statistic, so we're glad to see this. We also know the genome was 100% complete, and there was only a 0.17% contamination throughout the entire genome, so we are comfortable moving forward from here. Throughout our research, we identified many metabolic pathways, but narrowed it down. So our organism has genes involved in a complete cycle of glycolysis and citric acid cycle for our more basic carbon metabolic pathways. We're not quite able to identify a complete cycle of nitrogen or sulfur metabolism, which was something we were hoping to find. And only a few genes were missing in a complete denitrification pathway as well. We can get nitrite to nitrous oxide, but cannot fully convert it from nitrate to nitrogen itself. And kind of tapping into our first research question here, amino acids seem to be the major carbon and energy source that this organism relies on. And these 9 amino acids here, they were required for our organism to grow. So, in a lab setting, we had to supplement these amino acids to our organism for it to continue to grow and live. And through other metabolic pathways this organism has, it was able to synthesize any other amino acids it needed to continue its life. And these 9 heme binding motifs were of interest to us, so we could better understand our organism's potential involvement with extracellular electron transfer that can be observed in electrode oxidation. So 8 of the 9 were localized in the cytoplasm. In this top one here, it didn't really give us an exact answer of where it was localized, and we found these results through a place called PSORT. We definitely want to extend a special thank you to Dr. Annette Rowe and Joshua Sackett and the University of Cincinnati for helping us through all of this and providing the information we needed to get the results that we now have. Thank you.